we are going to see the ultraviolet visible spectrophotometer or UV visible spectrophotometer. The content of this video: UV visible spectrophotometer, principle of UV visible spectrophotometer, instrumentation of UV visible spectrophotometer, working principle of UV visible spectrophotometer, application of UV visible spectrophotometer, and finally advantage and disadvantage of UV visible spectrophotometer. The introduction of UV visible spectrophotometer. The spectrophotometer is an instrument for measuring the transmittance or absorbance of a sample as a function of the wavelength of electromagnetic radiation. The spectrophotometer technique are mostly used for quantitative analysis of molecules or ionic species in solution. This spectrophotometer generally consists of two different devices. One is spectrometer and another one is photometer. The spectrometer are used emits light of the desired wavelength, and photometer are used detect the amount of light absorbed by the solution. The principle of spectrophotometer. The principle is UV visible spectroscopy is based on the interaction between light and matter. When the light passes through or is absorbed by a molecule, it can cause the molecule to vibrate. The wavelength of light that is most strongly absorbed by a molecule is called the absorption maximum. The instrumentation of spectrophotometer includes light source, wavelength selector, sample container, monochromator, amplifier, detector, data recorder. The first one, light source. A UV visible spectrophotometer typically used two light sources. One, deuterium lamp used for ultraviolet range, and tungsten halogen lamp used for visible range. Deuterium lamp is a gas discharge lamp that emits light in the near ultraviolet and ultraviolet region. Tungsten halogen lamp, the lamp that emits light over part of the UV spectrum and over the entire visible range. And second one, wavelength selector. The wavelength range used for UV visible spectrophotometer is typically 190 to 900 nanometer. UV range 100 to 400 nanometer and visible range for 400 to 800 nanometer. Third one, sample container. The material or sample holder is made from also have a UV visible absorbance cutoff. Glass will absorb all of the light higher in energy starting at the 300 nanometer. Quit will be more practical as the absorbance cutoff is around 160 nanometer. And fourth one, detectors. The detectors are used to measure the transmitted or reflected light from a sample and converted it into a signal. Photomultiplier tube are used, one of the most sensitive light detector made. Charge coupled device used for multiple wavelength simultaneously. And fifth one, the monochromator is a key component of ultraviolet visible spectrophotometer that are select a specific wavelength for an experiment. The monochromator are usually located between the light source and the sample. Amplifier. The amplifier is a ultraviolet visible spectrophotometer amplifier. The low intensity signals generated by the photocells to create clear recordable signals. And finally, data recorder. The data recorder is a ultraviolet visible spectrophotometer records the transmission or absorbance of light to each wavelength. Working mechanism of spectrophotometer. The spectrophotometer should be turned on 10 to 15 minutes before use. The light source emits a spectrum of light and the monochromator select a specific wavelength. The selected light pass through the sample cell and the detector measure the intensity of the transmitted light. And finally, the readout displays the absorption of or the transmitted walls values. Application of spectrophotometer used chemical analysis. How a sample absorbs, transmits, and reflects light to quantify and qualify chemical components. Biomolecules analysis can accurately determine the concentration of protein, nucleic acid, and other biomolecules. Used pharmaceutical research can identify and quantify components in pharmaceutical products to ensure their quality and efficiency. Purity testing can access the purity of DNA samples, which is important for downstream application like PCR and DNA sequencing. Quality control used in industries like food, cosmetics, and pharmaceutical industries to ensure the quality and consistency of product. Trace metal analysis used for determine the trace metal content of this trace metal steel. Sunscreen testing can analyze sunscreen formation to ensure they absorb or reflect ultraviolet radiation to protect the skin. Advantage of spectrophotometer. This is simple and inexpensive experiment. Most organic molecules absorb ultraviolet visible light. This is quick measurement, easy to use, analyzes organic and inorganic components. And finally, disadvantage of spectrophotometer. Cannot analyze all components. Calibration process is lengthy. Stray light, low sensitivity and selectivity. And finally, end of the video. Thanks for watching. Thank you.